Antenna at 680 nanometer is not part of photosystem 2 because antenna pigments in photosystem 2 absorb light at shorter wavelengths and transfer energy to the reaction center P680. The reaction center is a complex of proteins and pigments. The subsequent electron transport involves several protein complexes that contains enzymes or are enzymatic in function, facilitating the redox reactions. Non-cyclic photophosphorylation requires both photosystem 2 and photosystem 1. Photosystem 2 is found only in grana thylakoids. NADPH is synthesized only in granule thylakoids and not in stromal thylakoid. Cyclic flow of electron can lead to synthesis of ATP only. The water splitting complex is located on the inner side of the thylakoid membrane, but only in photosystem 2, which is present in the grana thylakoids. The oxygen evolving complex of photosystem 2 contains Mn2 plus ions and Cl minus ions that are essential for splitting water during photolysis. Stromal lamella contain only photosystem 1. Cyclic photophosphorylation occurs. In stromal lamella, only hydrogen carrier, that is plastoquinone, can create proton gradient. When light more than 680 nanometer, protons are pumped into both granule and stromal thylakoid lumen, decreasing their pH compared to the stroma. NADP reductase enzyme is present on the stromal side of granule thylakoid membrane. Prokaryotes like cyanobacteria have only photosystem 1 and lack photosystem 2, hence photolysis of water does not occur. Protons or hydrogen ions produced by photolysis of water accumulate in the lumen of thylakoid because the splitting of water is associated with the photosystem 2. At low light intensity, light becomes the limiting factor for photosynthesis, not CO2 concentration. So increasing CO2 will have no effect on the rate of photosynthesis. Oxaloacetic acid is 4-carbon acid formed in C4 plant as first fixation product. Photorespiration does not occur in C4 plants. In C4 plants, rubisco is absent in mesophyll cells and present only in bundle sheath cells. Both mesophyll cells of C3 plants and bundle sheath cells of C4 plants have rubisco, contain stromal thylakoid, and can perform cyclic ATP synthesis. Bundle sheath cells in C4 plants generally have more and larger chloroplasts than mesophyll cells. Photosystem 2 is absent in bundle sheath cell but photosystem 1 present in C4 plant. Photosystem 2 only present in granule thylakoid in mesophyll cell of C4 plant. Kranz anatomy in C4 plant features two types of chloroplasts, mesophyll cells, bundle sheath cells. Compensation point for light is the time when photosynthesis equals respiration, and it typically occurs twice a day in the morning and evening. At low light intensity, the rate of photosynthesis mainly depends on the amount, intensity of light available. Light saturation occurs at 10% of the full sunlight, hence except for plants in shade or in dense forests, light is rarely a limiting factor in nature. If light intensity is low, increasing the CO2 concentration will not significantly increase the photosynthetic rate. If the temperature is low, the rate of enzyme activity like rubisco will be reduced, decreasing photosynthesis.